everyone. Today in this video, I will be continuing with the chapter photosynthesis. In this video, I am going to discuss on the experiments that are related to photosynthesis. Do watch the video till the end. Do not forget to like, share and subscribe to my channel. Now let's begin. Before performing the experiments on photosynthesis, we need to perform one primary step, that is the discharging of leaf. What does discharging of leaf mean? Discharging of the leaf is nothing but it means the removal of the starch from the plant. Why it is so important? It is so important because if the starch is already present in the leaf, before performing and after performing the experiment, we cannot conclude whether whether the photosynthesis has taken place or not, or we cannot draw an conclusion from this whether the factors are how these factors are helping to perform the phenomena of photosynthesis. So to discharge a leaf or to performing the discharging of a leaf, what we have to do, we have to keep the plant initially placed in the dark for 24 to 48 hours. If we keep the plant or we keep the setup in the dark for 24 to 48 hours, the whatever starch is present, that is utilized by the plant for the survival. This ensures that when we perform the experiment, there is no preformed starch present in the leaf. The next thing is that how will we understand whether the plant has performed photosynthesis? This is drawn or this is understood by iodine test, which is a second to perform iodine test. What we have to do, we have to do few steps we need to perform to conclude whether the starch is present or not, or to check the presence of the starch. What we have to do first, we have to keep a leaf or we have to dip a leaf in a boiling water. What does happens if we keep the leaf in the boiling water? This kills the cell. Then what we do, we boil the leaf again in the methylated spirit or any alcohol. What does this do? This removes the chlorophyll and the leaves become a pale white color. Obviously, the green color was due to the presence of chlorophyll. So, on boiling the leaf in the presence of methylated spirit, the chlorophyll is lost or chlorophyll is removed. Then what we do, we again wash it in a normal water and place it on the surface. Then we add few drops of the iodine solution to it, and the presence of starch is indicated by the blue-black color. Means those regions which have starch will show a blue-black color, and those regions where there is no starch or there is a starch has not been formed at that area or in that region will show a brown color, which is the color of the iodine. So now let's begin. There are four experiments that are related to photosynthesis. Number 1, to show that chlorophyll is necessary for photosynthesis. Number 2, to show the sunlight is necessary for photosynthesis. And the third, that is CO2 is necessary for, that is carbon dioxide is necessary for the photosynthesis. And the fourth is the fourth experiment is related is based on that oxygen is evolved during photosynthesis. So let's begin with experiment number 1. So what we have to do, as I have discussed earlier, first we need to discharge the plant. After the discharging the plant, what we'll do, we have taken a variegated leaves. What does variegated leaves mean? Variegated leaves actually means that in some areas chlorophyll is present and in some areas chlorophyll is not present. That means if I have a leaf, it is spreaded in unequal form, means this circle region shows no chlorophyll and the rest of the leaf shows presence of the chlorophyll. So the example of such leaf is Croton. This is a common question. Give an example of the variegated leaves. So please remember the answer, that is Croton. Then we have taken a variegated leaf, which was Croton. We have discharged the leaf, and we have seen how to discharge a leaf by keeping it in the dark for 22 to 48 hours. Then what we have done, after 48 hours, we have exposed the plant to light for few hours, like for 3 to 4 hours, and then we have taken the leaf from that plant and we have performed the iodine test. On the leaf, this was the variegated leaf which was exposed to sunlight, and then after the exposure to sunlight, what we did, we added iodine. What will we see, that if wherever the chlorophyll is present, that means over here the color turns to be blue black, and the portion which is non-grey, or the portion which does not contain chlorophyll, is yellow in color or brown in color also, we can say. So we can conclude from this that only in those parts of the leaf where chlorophyll was present, the starch is formed in that region, and in the variegated leaf where chlorophyll is absent, there is no starch formed by the process of photosynthesis. The next experiment, which is to show the sunlight is necessary for photosynthesis. Similarly, the most important step over here first is to discharge the leaf, that is to the removal of the removal of the starch from the leaf. Then what we did, we have covered a leaf with a black paper. A portion of the leaf is covered with a black paper. You can cut a star on it, or you can keep without any doing designs on that paper. Now after this setup will be kept in sunlight for few hours. What we'll observe that, and then we'll take the leaf which is covered with paper. So suppose this is a leaf which was covered with paper. After the exposure to the sunlight, then what we'll do, we'll remove that black paper because it was attached with a clip. We'll remove the black paper and add iodine. Let me mark it with a dotted line to show this region was covered with a black paper. Iodine is added. What will happen? The portion of the leaf where the black paper was, the leaf which was covered with the black paper, show brown color, and the rest of the leaf showed blue-black color. This means that the portion which was covered with black did not receive sunlight and hence did not perform the process of photosynthesis, so no starch was formed. But those leaves which was not covered with the black paper were able to absorb sunlight, so there was activation of chlorophyll, and so there was the process of photosynthesis that has taken place, and the starch was formed there, which was indicated by the change of the color upon addition of the iodine. So we can conclude that sunlight is also essential for photosynthesis to take place. The next experiment that we are going to discuss is to show that carbon dioxide is necessary for photosynthesis. So what we have done, we have taken a discharged leaf. After the discharge of the leaf is for discharge, we have modified the setup. This is little what to say, this is a little modification from the rest of the experiment. The modification is that a part of the leaf was kept inside a flask which contained potassium hydroxide, or KOH, the formula of potassium hydroxide. So what we'll do, after putting a part of the leaf inside the flask and closing it with a cork and exposing the whole setup to sunlight for few hours, what we'll observe that the part of the leaf which was inside KOH shows no change on addition of the iodine. Why so? Because the carbon dioxide which was there in the atmosphere and which reached the stomata was all absorbed by the potassium hydroxide. So the role of potassium hydroxide over here is to absorb the CO2, and the leaf which was not up inside the flask perform photosynthesis, and they show a characteristic change on addition of iodine. So we see that CO2 is also essential for photosynthesis to take place. So very important thing which you need to keep in mind, that in question, in the experimental based question, they always ask the board always asks a question, what is the role of KOH in this setup? The role of KOH is to absorb the CO2. So when this setup is kept in sunlight, the carbon dioxide is all absorbed by the potassium flask kept in the flask, and so that leaf gets no CO2 and thus it cannot perform photosynthesis. Rather, the part which is not kept in the flask can absorb sunlight, can absorb photosynthesis, have chlorophyll, and they have all the essential thing which is necessary for photosynthesis, and they perform it, and they have starch in them. So which on upon addition of iodine is indicated by color change to blue black. So till now we have discussed three experiments. Number one was to show that chlorophyll is essential for photosynthesis. Second experiment was to show that sunlight is necessary. And third experiment was to show carbon dioxide is necessary for photosynthesis. This till now we have discussed what is all necessary. What are the main elements we need for the process of photosynthesis to take place. Now we are going to discuss about the experiment to show that oxygen is necessary or evolved during photosynthesis. Okay, this was a byproduct. If we remember the equation, which was CO2 plus H2O in presence of light and chlorophyll, gives C6H12O6, which is glucose, plus oxygen plus H2O. I have not balanced the equation. I have just represented it in the simplest form. So we have seen that oxygen was released as a byproduct. How to prove that and what we did? This was a very simple experiment. We have taken a flask which was filled with water. We have taken a water that is aquatic plant, that is hydrilla. The hydrilla, the setup was done, so the flask we have kept the inverted flask, conical flask, inside the hydrilla. That means when hydrilla was taken and the conical flask was placed such that the hydrilla was inside and the tip of the flask was pointed upward. The tip of the flask was then covered with a test tube, and the whole setup was carefully kept under sunlight for few hours. What we saw that hydrilla or any aquatic plant, what they do, they evolve oxygen in water. So this presence of oxygen can be seen as a droplets or as vapors in this test tube. This presence of vapors or bubbles basically indicates that oxygen is evolved during photosynthesis. So this again I am repeating the setup. We have taken a flask, we have taken a beaker filled with water, we have taken a hydrilla plant which was covered with a conical flask, sorry, funnel. The funnel was then covered with the mouth of the funnel was covered with the test tube, and when this setup was kept in the sunlight, it was observed that as the hydrilla plant release out oxygen, the presence of the oxygen was seen in the test tube as bubbles. So this indicates that oxygen is evolved during the process of photosynthesis. So this was all about the experiment that we need to discuss, and all that comes in your board examination. Do study this experiment very properly. As you can score good marks from it. Study the definition of discharging, how to discharge a plant, and remember that whenever we have to prove that this carbon dioxide, chlorophyll, and sunlight is necessary for photosynthesis, we had to discharge the plant. But when we have to demonstrate that the green plants release oxygen during the process of photosynthesis, in that time we have not discharged the plant. So this is the key difference between the elements needed for the photosynthesis and to prove what is released as a byproduct of the process of photosynthesis. Please do watch this video properly. If you have any doubts, do write in the comment section. Thank you.